Hi, everyone. I'm Gabe Prado, a wet lab FAS here at PacBio, and today I have the joy to talk to you about how PacBio has got you covered from extraction to sequencing on one of our Revio systems. So in this presentation, we'll be talking about the extraction of high molecular weight DNA, which is optimal for long-read sequencing, the positive correlation that exists between sample quality and sequencing performance, new whole genome and microlab prep method kits that will scale with your needs, whether you be low throughput, mid throughput, or a high throughput institution, and our compatibility partners who enable sequencing success, all in all with the goal of achieving customizable end-to-end solutions. So words are great, but pictures are better. Here at the bottom, we have a nice schematic demonstrating how PacBio enables your sequencing success, starting all the way with the DNA extraction with one of our nanobind kits on the left-hand side here, then going into the use of a short read eliminator, what we would call the SRE kit, how you can then go into library prep, what we would call SmartBell prep kits, sequencing onto a Revio system, and then the downstream analysis. So here's our first glimpse of data. This is data demonstrating both the extraction yields and hi-fi yields that came out of a variety of sample types with the nanobind pan DNA kit. You can see here with some relatively modest amounts of input material, DNA yields were great, and especially the mode sizes were very long. All in all, that after library prep, there were just these excellent hi-fi yields achieved at great median QV values. So let's talk about how one would go about evaluating DNA quality to make sure that it is of the best quality for long read sequencing. So we would recommend the use of the Agilent femtopulse system or another pulsed field gel electrophoresis system, all for accurate DNA sizing. I can't stress this enough. High quality, high molecular weight DNA, aka that which has long insert lengths, long fragments, that is critical for really achieving great and high data yields. Low quality or degraded damaged DNA is going to result in shorter read lengths, lower data yields, and lower library construction yields. Furthermore, we would suggest the use of a nanodrop system or instrument for determining the purity of your DNA. You want to ensure that your input material is free of any secondary contaminants or metabolites that might interfere with library preparation or hi-fi sequencing. Again, high quality pure DNA, that's going to result in better data yield. And finally, we suggest the use of the qubit instrument for accurate quantitation of double-stranded DNA. So let's take a look at some examples of both really high quality extracted DNA and suboptimally extracted DNA. So here at the top, we're representing both the femtopulse, our recommendation, or an alternative, the fragment analyzer, also from Agilent. At this time, I will note that if these systems are unavailable, you can use a tape station for DNA sizing QC. I would just hazard all of you that caution should be used when analyzing samples above 10 kb, as the tape station loses accuracy above that range. Here on the left-hand side, we have a little table representing our recommendations for input quality when going into library prep. For example, when you're doing large genome, whole genome sequencing, we would recommend or really require you to come in with at least 50% of your DNA at or above 30 kb, and definitely 90% above 10 kb. And for small genomes, aka your microbes or your metagenome, here we would say 90% of your fragments to be at or above 7 kb. And this recommendation really exists to ensure that both the reagents and sequencing consumables that you're using for your library are being used most effectively. Now, I alluded to it earlier, so let's dive in a little deeper into both this lower quality suboptimal DNA that was extracted. You can see a genome quality number of only 7.2 and a mode of 22.6 kb, as opposed to this really wonderfully sharp peak of high molecular weight DNA at 146 kb. This is what those two DNA inputs then went on to look like in sequencing. You can see that the more suboptimal input, despite it was still sequenced, and it still generated data. However, you can see the mean hi-fi read length was just over 12 kb, not the most impressive data yield, as opposed to that really high quality, ideal DNA extraction, where you have a mean hi-fi read length of about 22 kb. And for those of you who have sequenced before with our technology, you're likely aware that this shape right here of the read length distribution plot, this is exactly what we want with this defined, almost bell curve-like plot at or about 20 kb with maybe a small camel's hump on the left-hand side. Again, cannot stress this enough. High molecular weight DNA, that's really going to enhance your data quality. So at this time, I'm going to shift gears a little bit and talk about one of our newest solutions that we provide here at PacBio. This is the Revio Hi-Fi Prep Kit 96. This is an excellent solution for those of you out there who are interested in really high throughput solutions. This is a PacBio qualified method available on the Hamilton NGS Star MOA, and it's going to have automated solutions all the way from high throughput short read eliminator to high PET shearing of your high quality, high molecular weight DNA into the actual library prep. You can, for those of you familiar with the protocol, these are all the same steps, just done for you by the system. And finally, it also does the annealing of the sequencing primer, the binding of the sequencing polymerase, and that necessary cleanup, basically taking you all the way up until the point where you're ready to load onto a Revio. This is a great scalable solution at every step of the workflow. It brings down 40% of the workflow costs thanks to that pipette shearing, and it is a 13-hour automated workflow for 96 samples. Now, we want to make sure that you don't feel that this automated solution is going to lead to lower quality hi-fi yield. We want to reassure you that our automated solutions don't lead to a compromise in the quality. Here we have some genomic DNA that was extracted. You can see the GQN is only about 7.0. There is this pretty significant hump on the left-hand side of fragments between 1300 to 10 kb. Really these small DNA fragments that might gunk up your library prep and then lead to poorer hi-fi yield later on. Well, here's what it looks like after treatment with the SRE. You can see it in blue. There's really been this removal of the low fragments, and thus a bigger proportion of the population is made up of those really big, long fragments of DNA. The GQN has shot up. And finally, here is what that same SRE treated genomic DNA looks like post shearing in red. You can see that here. You can see that with the shearing with the Hamilton came down to about 18 kb, which is really that sweet spot that you want to be for optimal hi-fi yield generation. Now, again, we want to reassure you that there's no compromise on performance. This is what the hi-fi yields looked like for that very same library that we were just looking at the femto traces for. You can see hi-fi yield was over 120 gigabases, read mean lengths at about 17 KB. Truly wonderful looking hi-fi read length distribution plot here on the right. Again, nice bell curve, somewhere between 15 to 20 KB, only a small hump in the 5 KB range. Pretty much exactly what we're looking for in the ideal hi-fi run. Now there is a homolog to the Hi-Fi Prep 96 kit. This we call the Hi-Fi Plex Prep Kit 96. This is an excellent solution for those of you working with microbial whole genome sequencing, metagenome shotgun sequencing, or amplicon sequencing. The kit brings all the necessary materials for what we saw before, and the Smart Bell adapter index plate 96 so that you can multiplex your samples as you go. Overall, you can do up to 1,536 samples in a single Revio run. To clarify, that's 384 samples per smart cell, 4 smart cells running simultaneously on a Revio. This leads to a 50% reduction in per sample costs. You can do 96 samples in 6 hours. Note, there is a minimum of 24 plex that is supported for this, and you need a little less than 300 nanograms of DNA per sample. Again, this is another PacBio qualified method available on the Hamilton NGS Star MOA. Now, here is the big question. How much will it cost you? Well, we're happy to say that it's only going to be $39 per sample. Here's some data demonstrating the practicality of the Hi-Fi Plex Prep Kit 96. You can see that once again, you don't have to sacrifice data quality for scale. This is microbial DNA. You can see here that the median read length was about 12 KB. Your Hi-Fi reads yield was almost 90 gigabases. Really great performance for microbial samples, all done at a really high scale for those of you who have high throughput demands. Now, this is an excellent table that I would encourage many of you to either screenshot or keep record of, just so that you know that where your lab currently is in their needs or where you foresee being in the future someday, you can see what instruments and solutions PacBio already has present so that you can be empowered to meet your project needs. We've broken it down from low throughput, aka about 24 samples or less on the left-hand side, mid throughput, which would be 24 to 48 samples at a time, or high throughput, where you're looking at greater than 48 samples at a time. All of these solutions have scripts that you can find on the PacBio website, or you can contact your local FAS, field application scientist, who can help guide you to this information. Finally, I just want to close off by highlighting the PacBio compatible partner technology. Here we have highlighted various partners of ours who can meet you at individual steps along the workflow so that you are empowered in your sequencing all the way from extraction to secondary or tertiary analysis. And with that, I want to thank you all for your time. Please feel free to contact your local PacBio support team if you have any questions or concerns. Thank you all again for listening.